Well, the project was funded by the EU for the last 4 years. It's a so-called EID, European Industrial Doctorate, which means that we have PhD students, 5 in total, which spend part of their time in one of the companies that are included in the project and part of the time at the University of Potsdam.
There was a clause that we have to work in industry, so we have to have industrial experience along with our doctorate studies.
Most of the focus in research in a vegetable breeding company is focused on biotic stress and not abiotic stress, even though we see that abiotic stress is becoming more and more important worldwide. So therefore we decided to be a partner in this project.
For me, learning all these processes from basic till production was really important and really nice to get.
When you combine academia and business, you can really make a difference.
We produce biostimulants and specifically seaweed extracts. And one of the key issues there is our claims was that it could mitigate stress, but there was no proof that that was actually real. So we wanted access to see for definite whether extracts, seaweed extracts specifically, and specifically Super 50, could mitigate that abiotic stress.
That is very fascinating to me to not only do science, but also see it translating into a product or basically into an industry.
It was critically important because we're in the business of selling solutions, and specifically solutions to mitigate abiotic stress. Without sufficient proof, and sufficient proof at a molecular level, you do not have the confidence to sell those solutions. So that was why it was critical that we engage in this project.
Biostimulants could help for the sustainable crop improvements for the growers in EU and in all around the world.
So basically, we start out from, a defined number of model species or model crops. We grow them under defined conditions in a greenhouse or in phytochambers. We grow them until a certain developmental stage or different stages. We spray the biostimulant Super 50 to those plants once or twice, and then after some time we check the behavior of the plant.
In my project, I mainly work on 2 stresses. That's herbicide-induced oxidative stress and also drought stress. That means if I prime the plants with the seaweed extract, then it can protect the plants better from the stresses thereafter, for example, the drought stress or the oxidative stress.
And we saw that tomato plants and Arabidopsis plants treated with Super 50 behaved much better. The next is to look at the genes, and now we know which genes are responding. So we can, by looking at the genes, we can now somehow deduce the biochemical pathways that are affected by Super 50 or developmental pathways that are affected by Super 50 treatment.
What I do is I do a comparative analysis of the active oxygen species, and then I try to come up with are there any specific kind of reactive oxygen species genes.
And I perform an RNA-seq analysis, and with this analysis I could find genes that were highly upregulated so that the plants express it a lot, and other genes that were downregulated. And with these genes I could use it in the future, or ENSA can use it in the future to help the cultivated tomato, improving stresses like salt stress or drought stress.
Yeah, we also organized a conference on abiotic stress in plants. And I think the important thing is that plants are subject to multiple stresses. It's not just one stress like drought or heat. Very often there are different stresses that come together, and understanding the response of plants to these multiple stresses or recurrent stresses, this is very important for agriculture. And I think this is something we have to follow up in the future more intensively. And I think also out of the conference, we will have options for new joint projects.
Well, the training part in crop strengthen was really exciting. We trained from different lecture series and different conferences and also different workshops. So that training part in crop strengthen, that was really exciting. And I hope that will help me in future to become a successful researcher.
It's very important to apply what I learned or people got from their labs to the fields. I want to apply what I know to the near future for the farmers.
Benefits as per training, they were tremendous. So not only for the science aspect, so it's very important to have a good, effective communication between all the three partners, and not only towards science, but also organizing various things. I also went for a couple of conferences, presented my poster, and interacted with other people in the scientific community, and to build up, relations, probably for future collaborations, etc.
Apart from the research, another thing, because it was an industrial project, so I got to know that how to check what are the risks involved in the process, in the PhD projects, or in any kind of a project planning. What you have to do before you start the project, while you are into the project, and after the project.
I think that having a PhD student working dedicated on a project for several years, on a single project, is very much different than what we are used to.
You have an access to a source of highly intelligent, multicultural students in different aspects of biotechnology and bioinformatics who are striving to get their PhDs, but also striving to make new breakthroughs. So this is a fabulous combination of university, students, and business.